Hello there, Biol-50. Today we're talking about the senses. So let's get going. Oops. Okay, so senses, in terms of organs, there are general sense organs and special sense organs. Sorry about that. General sense organs exist as individual cells, or we can call them receptor units. In other words, they're just kind of their own little unit, and typically they're very widely distributed. And so an example of a general sense organ would be this Pacinian corpuscle that gives us a sensation of pressure in the skin as something pushes down on the skin. And we have Pacinian corpuscles all over our body, in addition to these Meissner's corpuscles, which give you a sensation of more gentle pressure, more gentle touch. And then the Raffinings ending that kind of give you touch pressure as well. And so there's a number of different receptors that you'll find in the epidermis that are sensitive to temperatures, pressure, and wind, actually, believe it or not. So the other type of sense organ is called a special sense organ. And what this is an organ that's really big relative to like a pressure receptor in your skin, like a Meissner's corpuscle. It's usually very complex, and it has lots of different tissues associated with it. So if we go back one slide, each one of these, Meissner's, Ruffini, Pacinian, even over here, well, free nerve endings are just free nerve endings. They're the dendrites of a nerve, right? These are all epithelial in nature, one tissue type. You come and look at the eye, you have connective tissue on the outside, you have muscle tissue associated with the eye. There's a lot of different tissues associated usually with special sense organs. So you have lots of groupings of specialized receptors in certain areas. We'll talk about that pretty soon. Over here, it's in the retina of the visual system. So we'll get to that in more detail. So let's start first talking about how we classify sense organs. Basically, you have the special or general. Okay, we did that. You can also classify them based in terms of whether or not they have a capsule around them or if it's a free naked nerve ending. An unencapsulated sense organ is usually going to help you sense pain, and that's what you have in your skin. Encapsulated starts going into more complex processing, though. The more complex the receptor, the more complex the processing of the information. So let's look at types of stimuli that activate the receptors in sense organs. All right, this is the way I like to think of classification. What type of energy is driving the receptors? So we have photoreceptors that respond to light, right? Makes sense. Oops, let me get my pen going here. The photoreceptors respond to light. Chemoreceptors respond to chemicals in the environment, and that can consist of like being able to taste salt or sugar. Pain receptors, also called nociceptors, respond to injury. You don't have to bleed for an injury to happen too, right? Or necessarily see bleeding to realize that there's been damage to tissue. Thermoreceptors are sensitive to temperature change. Mechanoreceptors are sensitive to movement. So in other words, they can detect movement or deviation from center. And we'll talk about those in a little bit in the auditory system. And then proprioceptors. This is one that not a lot of people think about. But this tells us what our position of our body is at any given time. In other words, how long are your muscles? So if you are standing up right now and you have your arms hanging to your side, there are proprioceptors or stretch receptors in the muscles of your arms telling your brain, oh, your arms are hanging down to the side. Now, if you go and make a muscle, sun's out, guns out, guys, or everybody. If you make a muscle, then the proprioceptors are going to start firing differently, telling your brain, oh, I'm holding my hand up in kind of a muscle form, kind of a looking like as Arnold-y as I can get, okay? So those are proprioceptors, giving your brain an idea of where your body is in space. Now, how do you take a stimulus like pressure, proprioception, we just talked about, light energy, and convert it to sensation? This process is really important. It's called, well, how complicated do we want to get? It's called signal transduction. So you're transducing one form of energy, say light energy, into something that the brain can read, and that's electrical impulses. So all organs, all sensory organs, have all these common characteristics. They're able to detect a particular stimulus that they're associated with. So like your eyes, your retina are able to detect light. Your ears are able to detect pressure waves, actually, air pressure waves. We'll talk about that in a bit. Each sensation or each stimulus is converted from whatever the energy is, light energy, pressure energy, temperature energy, into electrical nerve impulses, okay? So let's say, for example, you have heat. That gets converted to action potentials, which are the same thing as a nerve impulse. The reason why we say an action potential, because that's a single cell firing off to the brain, sending electrical signals to the brain. So you're going from heat, converting it into electricity, which is pretty crazy. The nerve itself, the nerve impulse, is what's perceived as the sensation. It's not the actual heat. It's that transduction into the nerve impulse that your brain can see. So let's say, for example, you had thermoreceptors on your skin that weren't undergoing action potential propagation when you put something warm on your skin. Your body won't see it. Your brain won't see it. You won't detect it. That's exactly what happens to you when you go to the dentist and they give you the shot of lidocaine into your mouth to go in there and drill. It's not Novocaine anymore, guys. It's a new, more specific medication called lidocaine. When they inject the lidocaine into your mouth, if you're getting a filling done or something, what they're doing is they're blocking nerve transduction, nerve impulse generation and propagation at the level of the mouth. So what happens is they're drilling in there, but you can't feel it because that nerve impulse has been knocked out. So in essence, you're not able to perceive that sensation because it got knocked out. So perception, I guess you could think, is a malleable experience depending on whether or not the receptor is working. And if the receptor is working, is the receptor sending the information to the brain appropriately? And if it gets to the brain, is the brain processing it appropriately? So perception is an interesting idea. Is it all sensation? Well, you can look things up. Maybe at the end of this lecture, I can point out some kind of interesting things to you guys. If you're interested, send me an email. How about that? Send me an email if you're interested in books on sensory perception and how to trick people. Magicians do it all the time. Okay, let's go back to our general sense organs. General sense organs, typically in the skin and the epidermis, consist of our free nerve endings, which pretty much tell you about pain, right? Paper cut, you're going to feel it right here, all right? The free nerve endings exposed to oxygen tell your brain, hey, guess what? You just made a lesion in the skin. It can also tell you about crude touch. When we say crude touch, it's just like, it's not fine detection of touch. It's just, hey, something's on your arm. But you don't know what it is. The Meissner's corpuscles right here, kind of more superficial in the skin, give you an idea of fine touch and vibration. In other words, if you have a mosquito, okay, here's my idea of a mosquito. Blech, I hate these things. Isn't that ugly? Oh my gosh, it needs a big eye. Yeah, I didn't go to art school. I went to neuroscience graduate school. What can I say? Okay, that's my mosquito. As the mosquito is walking along your skin, you may not actually feel it because it's so lightweight. But once it injects saliva through its proboscis under your skin, you might feel that. You might feel the little poke. And a lot of that has to do with either activation of free nerve endings or pressure on the Meissner's corpuscles. Okay, so that's light touch, fine touch, vibration as a mosquito might walk past the hairs on your arm. The Ruffini's corpuscles over here, deeper, have to do with touch and pressure. Typically not super strong pressure though, but it's definitely a deeper pressure than a mosquito walking on your skin. A mosquito wouldn't activate a Ruffini's corpuscle or a Pacinian corpuscle. These are stronger pressure. Okay, so this is hard pressure. This is, Ruffini's is more like medium pressure. And the Meissner's are light light pressure, okay? Now, the Krause's endings up here, they have to do with touch as well, but also touch and movement of the skin. That's all we need to say about that. Golgi tendon organs, which are not in this picture, have information about proprioception. You'll find Golgi tendon organs associated with muscle, and that includes the muscle that might be associated with hair. And then muscle spindles, also proprioception, but we don't have that in here. Skeletal muscle would be the muscle spindles. Now, special sense organs, the one that we always talk about pretty much first of all, is the eye, because vision is really important to humans, really important to most mammals, actually. We see the world in color as mammals. Some people have green-red color deficiency. Don't worry about that right now. The eye itself is structured like a camera. In fact, we call it the camera eye, all right? At least an old description is. But let's look at the layers of the eye to start with and then describe how the eye perceives light. So the outside of the eye, the white of the eye, is called the sclera, and this is a really tough layer of basically connective tissue. The cornea is the clear area overlying the iris and the color, okay? So the cornea is transparent. It's the transparent part of the sclera. It's just basically not white. The choroid is the colored vascular area. It keeps light from basically scattering. It makes things look less, let's put it this way, it reduces glare. The color prevents light scattering from happening. The color actually absorbs light. So the front part of the eye is all about controlling how much light gets in through the pupil, all right? The ciliary muscles are associated with setting the diameter of the pupil, and we'll see a better representation of ciliary muscles from the inside of the eye, okay? The pupil itself is the hole in the middle. The iris, like I said, is the color. And then the contraction of the iris, this region right here, right outside the pupil, is what dilates or constricts the pupil. Dilation is making the pupil larger, and constriction is making it smaller. The retina is on the inside of the eye. You can't see it here. This is what contains the rods and the cones, which are necessary for seeing the world around us. Rods detect black and white, or color intensity, and the cones are receptors that detect color. They're really important for day vision, and that comes in 3 flavors: red, green, and blue, all right? Everything that's not red, green, and blue is your brain has to calculate that color. So pink is a combination of red and blue being activated, all right? And so we'll talk about perception of color in a little bit, just a little bit. The conjunctiva is a mucous membrane right down in here, kind of near the inner corner of your eye. And you can't see it unless you pull your lower eyelid back. But the conjunctiva is a mucous membrane that makes mucus, and it covers the sclera and it lines the eyelid, and it keeps everything nice and moist. It produces mucus that'll trap bacteria. It'll produce mucus that can trap dust, stuff from getting in your eye. And then the mucus plus tears, which are made by lacrimal glands, will get washed down through the lacrimal ducts. And you have a duct up here and a duct right here. Usually you can't see the one on the top of your eye, but if you go home and you go look in the mirror, pull your top eyelid up a little bit and you'll see a little tiny pinhole right there. And that's one lacrimal duct that collects tears and takes the tears back into your sinus cavity. Now, last thing that you can't see here, but behind the pupil is the lens. The lens looks like a skittle, frankly. It looks kind of like a clear skittle, and it helps us focus the rays of light onto the retina. So here's a kind of a cross section of an artist's rendition of the eye. So we've been talking about these outer regions. Here's the sclera. Here's the cornea, right? The clear part of the sclera. Here is the lens. Like I said, it looks kind of like a skittle. Here is the iris right here. The ciliary muscles associated with the lens are right on the sides of the lens, right here and right here. So when our eye is trying to focus light on the retina, which is this back yellow layer here, the ciliary muscle will pull to the right or to the left to flatten the lens a little bit, and that focuses the light as it comes through the eye. When things are blurry and you squint a little bit, as you squint, it actually puts pressure on your eyeball, changing the tension of the ciliary muscles, allowing your lens to accommodate a little better. Unless you're my age, no amount of squinting is going to help me. I'm pretty much blind, but not blind as a bat. Bats have excellent vision at night, actually. But so that's how our eyes focus, is the ciliary muscle contracting or relaxing to change the shape of the lens so that as light goes through, the light focuses right here on the back of the retina, on the fovea. The fovea on the retina has the highest density of photoreceptors, both rods and cones, in the entire retina. The visual axis is the line of photon or light travel, okay? Now let's look at the chambers of the eye. We have the anterior chamber, which contains aqueous humor. Aqueous humor is just kind of a watery liquid. It helps maintain the pressure of the eye, helps keep the eye happy and healthy. The posterior chamber has a thicker fluid called vitreous humor. It's kind of much more gelatinous. The vitreous humor and the aqueous humor are produced, are produced by cells in the eye, and it helps maintain pressure in the eye. If pressure goes too high in the eye, then you can end up with a disease called glaucoma, and basically it damages photoreceptors and sensory structures in the eye. Basically, it can damage the lens, it can damage the retina itself. The vasculature can become damaged. It can be a mess. Over here is the lacrimal carbuncle. This is the inner kind of side of the eye that you always see. And here's a part of the lacrimal duct. Okay. So let's talk just a little bit about how we see what we see. I mentioned the rods and cones. So if we look at, let me go back one slide. So we're going to be looking at light traveling to the retina here. Okay, so this is the spot that we're going to be looking at. Let me get a different color because that's not working so well. Green I don't think would work. Let's just use black. We're going to be looking at this region right here, the fovea. Now, light would be coming in like this, okay? So that's light. Way back here in the wall of the eye is what we call the pigmented layer. Now, I didn't point this out in the last slide. Probably should have. Let me see if I have it there. Here's, oops. They don't really have it here, but it's behind the retina. The pigmented layer has cells that are full of melanin, and the pigmented layer is important for absorbing extra light in the eye so it doesn't bounce around back there, because you know how light loves to reflect on things. You have a series of photoreceptor cells. Now I'm going to show this picture blown up in a few minutes, okay? So the innermost layer right here has the photoreceptor rods and cones. The idea is that light comes in and goes past all these other cells and interacts with the rods and cones down here. The rods and cones send information to the bipolar cells, which then send information to the ganglion cells, which then send information to the brain via the optic nerve, okay? Now, the optic nerve itself generates what's called the blind spot, where the optic nerve and the vasculature come in and out of the eyeball. There are no photoreceptors because you basically have a bunch of tissue going in and out of the eyeball there. What's interesting is that if you look over here at the right eye, half of the right eye actually sends information to the right side of the brain, and half of it sends it over to the left side of the brain, and vice versa with the left eye. Right here at this crossover, we call this the optic chiasm. Chiasm means like X, okay? And so what happens is each eye sends information to its own cerebral hemisphere, ipsilateral, and it sends information to the opposite side, contralateral. Ipsi means same, contra means opposite. Information takes a first process pass over here in the lateral geniculate body, lateral geniculate nucleus on the right and left side, and then information is sent to visual cortex. The occipital lobe, as we remember from the last chapter, is where visual processing happens. Primary visual processing happens in the lateral geniculate. You don't need to worry about that. But basically, information from the eyes gets mapped to both sides of the brain. Why is that important? Well, we are very, very visual creatures, right? Especially predators. We need to be able to react to the environment very, very quickly and make very, very good visual estimations to undergo dangerous tasks. So let's say you are going after a warthog, okay? I don't know about you, but warthogs look kind of scary to me with their like teeth and all that stuff. If I'm going to go jump on a warthog, I'm going to want to do it right so that I don't get hurt. Well, the right and left eye are sending information to the brain, calculating how far do I need to jump. Also, you know, the frontal lobe is asking, are you sure you need to eat this warthog, right? So the brain uses information from both eyes to make decisions about the world around us. So here's a better picture of what the photoreceptors look like. The rods look long and the cones look short. Rods, again, you see black and white, okay? And the cones come in 3 flavors, even though they didn't show you here. Number 1 is red. You can see red with the red cones. Number 2, you can see green with the green cones. And number 3, you can see blue with the blue cones. Yellow, don't ask me about that. Now, if you have a certain type of color, or if an individual has a certain type of color blindness, I guess you would say, or color insufficiency, what that means is that on the X chromosome, the genes for the red and maybe even the green cones are damaged and you can't see red and green. All you see are shades of blue and kind of gray and maybe a little bit of yellowish, which is kind of crazy, right? Here's the incoming light passing through the ganglion cell layer, the bipolar cell layer, and into the photoreceptors. If light doesn't hit a photoreceptor, it just gets absorbed by the pigmented layer. And then here's our visual pathway again, which I suppose I should have written on this slide, but you can go on ahead and review the visual pathways, the decussation at the optic chiasm, and the ipsilateral projections on both the right and the left eyes into the right and left visual cortex. Okay, so how do we focus? That's called light refraction. Light refraction normally happens when the lens focuses whatever light you're seeing, like this item, the arrow, in an upside down and backwards format on the eyeball. So everything that you're actually seeing right now is upside down on your retina, okay? The brain takes this information and flips it right side up. Guess where? Right here. So it's upside down, leaving the eyes, and it gets flipped right back up by the time you get to visual cortex. Now, when you have myopia is the condition where the lens or the eyeball gets extra long and the lens can't relax enough to focus the image on the retina. So things look blurry. If you add a lens like a glasses or contact lenses, you can fix myopia, okay, or nearsightedness. Hyperopia happens when the lens can't, when the eyeball actually gets a little taller and the object is focused behind the retina, okay? The lens can't focus it and bring it right here in front of the yellow, okay? So then you add a different shaped lens, all right, to correct for farsightedness, which is hyperopia. So hyperopia is farsightedness. In other words, you can see far, but you can't see near. Myopia is nearsightedness. You can see up close, but you can't see far away, okay? Usually students in high school, you see many more people who have myopia than hyperopia. But by the time you get in your mid-40s, hyperopia is going to sneak up on you because for some reason our eyeballs like to get a little taller as we age. Don't know why. Okay, so disorders of special sense organs. We already talked about myopia, hyperopia. Astigmatism is a distortion in the lens or the shape of the cornea. And so instead of being a rounded cornea, you can have a cornea that actually looks kind of like a barbell shape. And what ends up happening when you have a barbell here like this is instead of a single arrow being projected back onto the retina, you actually have two focal paths, one that's here and one that goes through that area, and you end up with double vision. So astigmatism is a distortion of the cornea or lens, and you end up with double vision. And there are different degrees of that. Other disorders, conjunctivitis. This poor kid here. This poor kid has really bad pink eye. It's inflammation and infection of the conjunctiva. It can happen because of trachoma or chlamydial infection, acute bacterial conjunctivitis. This typically happens when kids get bacteria in the eye, or adults as well. Yeah, it's not pretty. You have to take antibiotic eye drops. And then conjunctivitis can also be caused by allergies. Strabismus is the improper alignment or cross-eyed. This can be corrected if you give this kid an eye patch here. I know this looks mean, right? To force the eye with the strabismus to start focusing out in the world. So this is a very common treatment. You just patch one eye and the other eye tends to recover. If this doesn't happen, it can cause permanent blindness because the one eye, the stronger eye, will take over the weaker eye, and the weaker eye just won't work as effectively. Disorders of the retina. The retina itself can detach. This usually happens just as you get older. It can be the result of a tumor or head trauma, hitting your head really hard. Diabetic neuropathy happens when individuals with diabetes type 1 or type 2 has abnormal growth in blood vessels. And that's not a good thing because that can lead to blindness, diabetic blindness. Glaucoma, like I mentioned, is a condition where you have pressure that decreases blood flow in the retina, killing the retinal cells. And then you have night blindness. I never see this right. Nyctalopia, yeah. And this is the inability to see in very dim light. And usually this is caused because you don't have a lot of vitamin A in your diet. Vitamin A comes from eating beta carotene, your carrots. When you eat carrots, let me check this out. This is amazing. When you eat your carrots, the pigments in the rods and the cones are synthesized better, I guess you could say. You get more pigments in there and you have more sensitive vision because you have enough vitamin A in your diet. Macular degeneration is basically where the retina starts degenerating. This is just basically, it used to be thought of as blindness in the elderly. And then red-green color blindness that I mentioned, or red-green color insufficiency, is where there's a mutation in both, either or both, the green and the red gene on the X chromosome that give rise to the pigments for red and green cones. It's pretty easy for a guy to have and to pass along, not quite as easy for females. Now, sometimes you have a situation where the optic nerve can start degenerating. That's usually secondary to other diseases like glaucoma or diabetes. And basically, if the optic nerve starts degenerating, then you won't see because the information is not being transduced from the eyeball to the brain. Sarcotoma is where you lose the central visual field because they've become damaged, but you can see laterally. And then a cerebrovascular accident or a stroke happens when usually in visual centers in the occipital lobe, but sometimes in the lateral geniculate nucleus. And that can be a situation where somebody starts out life totally normal, but as a consequence of a stroke, they might acquire color blindness. And it's called cortical color blindness because the cells in the cortex are no longer able to distinguish or perceive different colors, even though the eye is working just fine. So now let's go through the ear. Not go through the ear, but let's talk about hearing. The ear functions to do two things: allow us to hear and allow us to have balance. So the receptors for hearing and equilibrium, which is the same thing as balance, are called mechanoreceptors. Mechanoreceptors are mechano, or receptors that are mechanically activated. So let's first look at the different parts, or break up the ear into divisions. You have the external ear, the auricle, the pinna, the part that you see sticking up, that's the outer ear. The external auditory canal, your ear hole, which is about 1 inch in length. It contains lots of ceruminous glands, or, we've talked about ceruminous glands. It makes, cerumen or earwax, and it ends at the tympanic membrane. So looking at this outer, or the picture of the outer ear, this whole outer portion here is the pinna. Oops. Oh man. The pinna, the top is the helix, the triangular fossa, because it looks like a triangle, right? I like this, the antihelix. Okay. The concha. Concha, just so you know, always refers to kind of like this wrapping nature. The lobule, the targus, this little part here, the external auditory meatus, or auditory canal. When you come and take anatomy at the college, we do refer to this as the external auditory meatus. And then you have the antitargus over here. So targus, antitargus. Okay, so what actually starts transducing the information of hearing to our brain? This is a picture of the tympanic membrane, and the tympanic membrane is a thin piece of skin that's connected to bones on the inside of the ear canal. So the external auditory or acoustic meatus ends at the tympanic membrane, and the tympanic membrane then vibrates. It vibrates back and forth as sound waves, which are basically packets of air, bombard the tympanic membrane. Now, this tympanic membrane usually looks kind of like a little bit shiny. It looks shiny when it's healthy. If somebody has an inner ear infection, bacteria has built up behind this membrane, and pressure can build up, and it starts looking kind of foggy. It doesn't look like this gray, shiny, healthy membrane. It looks kind of dull. It may look like there's pressure behind it, and that's because as bacteria start building up in the internal space, which we'll see in a few minutes, that causes pressure on the tympanic membrane, which is very, very painful. So this is what we were just talking about. Here's the external auditory canal, external auditory meatus, the tympanic membrane right here, which is connected to the malleus, the incus, and the stapes. These are the auditory ossicles, or the bones. Now, as sound waves travel in, sound waves are packets of air that bombard the tympanic membrane, like I said. As the tympanic membrane vibrates back and forth because of the constant bombardment of sound, the ossicles start vibrating as well. The malleus rotates along by the incus, pulling the incus and the stapes back and forth, thus transducing motion into the cochlea. The cochlea is where we hear things, okay? The semicircular canals up here is balance. So any waves that are set up, or any vibration that's set up into the inner ear here, goes into the cochlea, not up into the semicircular canals, okay? So just be aware of that. The vestibular nerve and the cochlear nerve come together to form the acoustic nerve, okay, the 9th nerve, and this gives information to your brain about hearing and balance together, okay? So middle ear, this part right here, houses the auditory ossicles. It ends in the oval window, which is right here. The auditory or eustachian tube right here, the auditory tube, I always call it eustachian tube because that's what I was taught, connects the middle ear to the throat, okay? And so how do you get bacteria up here in the middle ear? Well, it comes in fluid coming in through your pharynx, or the back of your throat. So if you have bacteria in your mouth, which we all know we have bacteria in our mouth, right? That can travel up into the middle ear, set up residents in here, and those bacteria will start dividing because it's nice and warm in there, and there's moisture and stuff, and you'll end up, with an inner ear infection called otitis media. That's inflammation and an infection of the middle ear. So let's talk about the inner ear. Remember, this is the inner ear right here, okay? Hearing and balance. The bony labyrinth itself, which we'll see in a minute, is filled with a fluid called perilymph. And perilymph is kind of it has lots of sodium ions in it. It has lots of salts in it. The bony labyrinth is separated into the vestibule, semicircular canals, and the cochlea. The membranous labyrinth, on the other hand, has another fluid called endolymph. Again, another salty fluid, okay? Let's just go straight to what we're talking about here. Here is the separation between audition or hearing and balance, okay? So here in the cochlea, we have a membranous labyrinth, okay? Up here in the semicircular canals, all right, we have perilymph, okay? So down here, endolymph, perilymph. These little knobs at the bases of the semicircular canals are called vestibules and ampulla. Embedded in the vestibules and the ampulla and along the length of the membranous labyrinth of the cochlea are sensory hair cells. Now this is, let me see if I have a bigger picture. I do not. Sensory hair cells look like much more like this, okay? Now I know this because when I was, closer to your age, when I was an undergraduate, I worked in a lab that studied hearing. And what they studied were the hair cells inside of the cochlea. Okay, so underneath each one of these hair cells is a set of epithelial cells. See these guys? Supporting cells. And each one of these hair cells has on top of it, yes, what looks like little hairs. These are called stereocilia. Now what happens is this membrane up here, okay, the tectorial membrane, overlies all of these little hairs, okay? As sound is transduced from the tympanic membrane to the incus malleus and stapes, it gets transduced into the labyrinth here, which means you have vibration happening, vibration back and forth of this tectorial membrane. As that happens, it deflects the actual hairs on the hair cell back and forth as well. As you deflect in one direction, it causes, you got it, action potentials or nerve impulses to travel to the brain. Isn't that cool? All right. So in this way, you're able to transduce vibrational energy to action potentials or electrical energy in the brain. A similar process happens over here in the semicircular canals, except for instead of just having vibrations happening here, in the semicircular canals, you have that lovely perilymph. The perilymph flows around through these canals as we move our heads, okay? And as it swirls around, it offsets a similar organ with a similar set of hair cells in the vestibule and the ampulle, okay? When that happens, it tells our brain about rotational movement. In other words, is the fluid moving counterclockwise or is the fluid moving clockwise? Depending on the direction of movement, the hair cells, which look a whole lot like hair cells in the cochlea, right? I'm going to make an ugly hair cell picture here. Let's give it a membrane. If you have a similar process happening here, you have, oops, dang it. Sorry. If you have a similar process happening here, where you have the membrane being offset, going back and forth, that tells the brain which direction the fluid is going and basically which direction you're tipping your head. So it's a pretty amazing organ, okay? In the cochlea, this is called the organ of Corti, all right? Helps us hear. You can map tone, literally tone, like a note, to how far along a wave travels down the length of the cochlea. So when we come here, you see the cochlea as a snail-like structure. When you unwrap the snail here, you'll see that wave energy, that are longer wavelengths, it turns out, travel all the way to the tip. So you hear low sounds here, and you hear high-pitched sounds over at the beginning. So that's what the tuning is, okay? Low-pitched sounds at the tip of the cochlea, and then high-pitched sounds are detected by the base of the cochlea. And what ends up happening as we get older is there's always information coming through the high end, not as much through the low end, okay? This high energy eventually kills these cells, leading to age-related hearing loss. This information, the nerve impulses that are generated as a consequence of auditory action in the cochlea, is sent to auditory cortex, where it's interpreted as sound. It's pretty cool. So, what are some hearing disorders? You have conduction impairments. This basically stops information from going from the external ear into the inner ear. That can be lots of earwax or cerumen, and it can also be a tumor blocking the vibration of the bones in the middle ear. Otosclerosis is a bone disorder where the stapes is misshapen, and so eventually what happens is that sound is not conducted appropriately, so the vibrations aren't detected appropriately. So you first start hearing ringing of the ears, which is called tinnitus, and eventually it gets really, really loud and progresses to overall hearing loss. Otitis, again, ear inflammation caused usually by infection. You can have swelling or fluid in the ear, and that blocks conduction of sound as well. So that would be conductive hearing loss. Nerve impairments, presbycusis, progressive nerve deafness. This is just part of what happens as we get older. Progressive nerve deafness can also result from exposure to really loud sounds. So if you're around, this happens a lot, drummers in rock bands, drummers and bass players, those guys always end up with hearing deficits. Meniere's disease is a chronic disorder where individuals get tinnitus. They can also get a lot of vertigo as a consequence of Meniere's disease. Now, another special sense organ, which is really kind of entertaining, is taste, gustation. The receptors in the taste buds are chemoreceptors, yay! And what we taste usually is sweet, sour, bitter, and salty. There's another one called umami. It tastes like amino acids. But the whole idea is that it's important to know what you're putting in your body, right? And if it's something good for you, it'll probably, and if it carries lots of energy, it'll probably taste sweet. Bitter is usually an indicator of something not so good for you, even though coffee, we love it, right? And salty is also an essential flavor because we need salt. We need sodium chloride in our body to do certain jobs. The cranial, 7th and 9th cranial nerves take taste or gustatory impulses to the brain, and gustatory and olfactory work together to give us an idea of the flavor of something, right? So taste receptors themselves occur on our tongue, okay? Here are the tonsils back here. We have the circumvallate papillae, and then in regions adjacent to the edge of the tongue, you'll find taste buds. Taste buds are basically these little gustatory cells inside of a little pore with little hairs. And so we taste whatever the item is, let's say some glucose here. It'll lodge onto these taste hairs, stimulating these cells, allowing these cells to tell nerve fibers, ooh, I taste a Snickers bar or whatever. Now, that's just giving you an idea. Actually, it doesn't even give you an idea of the Snickers bar, actually. What it tells you is that you're tasting sweet, sour, bitter, or salty. Now, I don't know how long this lecture is going to be posted for or how many years I'll be using this lecture, but this is 2021, and we've just lived through COVID-19. People are just now starting to get vaccinated. People are going to be venturing out in the world again. It's great, right? We made it. We're almost there. One of the weird parts of COVID-19 is that people lose their sense of smell and taste. Why is that? Is it because these special sense organs are having cell death, right? Is it because these gustatory cells are dying. Well, it turns out no. You can still taste sweet, sour, bitter, salty, but it's just that. It just tastes sweet. It just tastes salty, okay? You don't know what you're actually eating. So if somebody were to put a potato chip on your tongue, you wouldn't know it was a potato chip. It would just taste salty. Part of what's going on is receptors in the olfactory mucosa become blocked by lots and lots of mucus. They actually don't die, which is interesting. They figured that out. Olfactory receptors are super, super sensitive, but they are very, very easily fatigued. In other words, they can get blocked up with lots of mucus and you won't smell anything. So eating a potato chip, tasting a potato chip, a lot of what you're actually tasting is the smell. It's the sensation of the smell that your brain is detecting as potato chip. It's the salt on your tongue that tells you salty, but it's the chemoreception of your olfactory receptors that gives you a better idea of what you're tasting. So next time you guys are healthy and you don't want to taste something that you don't like to eat, pinch your nose, breathe only through your mouth, and it will downregulate more than at least half of the flavor that you don't like. Odor-causing chemicals actually initiate the signal right here. So here's an olfactory cell, all right, that has little receptors dangling down into the olfactory, up into the upper nasal cavity. This is the cribriform plate, right? So the little axons are, or I guess it's not even axons, it's cell dendrites hanging down into the upper region of the nasal sinuses. We eat something, and as we breathe, some of the molecules come up here to the olfactory bulb, and because a lot of these are gas molecules that are water soluble, it activates these olfactory receptor cells and sends a signal to your thalamus saying, that was yummy. You know, this color of candy, doesn't look very good to me. If that was red, it would be, much better. Cherry flavor, strawberry. So that's it for our lecture on the senses, and I will catch you next time.